Hello, Professor.
Oh, hello.
Yes, sir. Please start the class. It's like
Okay, so let's get started. So
okay.
today's topic is about spectroscopic deep learning, particularly application in biomedicine. Before we talk about the deep learning method, we first look at the spectroscopy technology in biomedicine. Okay. We know that in biomedicine, we typically need to check the quality of the foods or any kind of like a herbal medicine or those kind of things that we use. And we also need to check up our health conditions, particularly diagnosis of diseases. And these tasks could be facilitated by spectroscopic technologies, like the examples of the on the left-hand side. The quality of foods actually dependent on the materials and microbes and those kind of things are. And for instance, if there are some toxic materials and the food, certainly it affects the quality of the food. If there are some harmful microbes, it can actually cause some kind of disease in your body. So this kind of the state of the molecules and also the state of the microbes. Of course, microbes are composed of molecules. So these states can be detectable by, for instance, infrared spectroscopy, IR spectroscopy. Uh, in medical diagnosis, it is similar to the quality of food because the health condition is also concerned with certain kind of molecules, proteins, genetic influence, those kind of things. Those kind of variations, molecular variations, can also be detected by spectroscopy, particularly Raman spectroscopy. So now we look at the specific use of the spectroscopy technology. One example is the near infrared spectroscopy. It is particularly good for measuring the chemical components and also essentially the composition of the that make up the material like the food, for instance, wheat flour. You can typically use near infrared spectroscopy to do that. And also regarding to food, for instance, the kind of drink, either alcoholic drink or food drink. So, so typically, they also have certain kind of molecules that actually determine the quality of this kind of liquid food. So for this kind of food, the molecular state can be measurable by a Raman spectroscopy. So the Raman spectroscopy is the kind of laser beam that the laser beam actually is directed at certain objects, and then the laser photons are absorbed by the molecules in the material, and then it excites the electron into the higher energy state, and those electrons can be reduced to lower energy state, thereby emitting refracted photons. Those photons actually form this kind of spectroscopy pattern. So this pattern definitely can be learned and recognized by an AI type of architecture. So another application is the quality of medicinal herbs, medicinal plants. We know that in medicinal plants, the amount of the active ingredients, they are very, very important. Those ingredients, they are molecules, right? So definitely we can apply spectroscopic methods, particularly Raman spectroscopy, for the detection of the molecular contents inside these kind of herbs. So how the Raman spectroscopy works, how it works uh, actually, I just described that, right? I already gave you some information about how it works, so I don't have to repeat it. But so obviously, different type of materials, be it herb or some kind of foods, they tend to produce different patterns of the Raman spectrum. So this Raman spectrum is actually in terms of the shift of the uh, 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 emitted light. Remember that the materials absorb the laser beam, laser light, the photons, and then re-emit the photons at a changed frequency. So that's why there's a shift. So Raman spectroscopy actually measures the shift of the emitted photons. So that's uh, uh, essentially that's how the Raman spectroscopy works. Another application of Raman spectroscopy is the detection of the state, the conditions of cells. It could be tumor cells. So tumor is a state different from normal cells, or it could be for instance, the stem cells. So stem cells can be potentially utilized for instance, tissue repair, for the anti-aging purposes, those kind of applications. Obviously, the quality of a stem cell is very important. So these actually are concerned with the state, the conditions of the cells. Obviously, they are dependent on the expressed molecules, the proteins in the cells or on the surface of the cells. So that kind of state can be measurable by Raman spectroscopy, Raman spectroscopy, those kind of techniques. So you could also uh, combine Raman spectroscopy with medical image. So you have this Raman image type of tools for the diagnosis purposes. So now the question is how can we apply the spectroscopy technique for the development of a deep learning model. So this is a example of such applications. Essentially, it is to measure the uh, so use Raman spectroscopy to measure whether or not there's a tumor developed in the brain. So that kind of applications. So you have some kind of probe inserted into the brain tissue, and then you can have this Raman spectroscopy light, and then the collection of the refracted light, you get this kind of Raman shift pattern. So the question is, which pattern actually corresponds to a cancer situation, which is a normal situation. So in this case, you can develop an AI. So essentially, this is a unit platform to actually read about this kind of Raman shifted data, and then to predict uh, the status, whether it is a cancer or it is a non-cancer. So this is one example of the Raman spectroscopy deep learning model for brain tumor detection, this kind of approaches. Nowadays, the Raman technology, the hardware, Raman hardware, develops very, very fast. So first of all, you need to have this kind of probe that can actually be inserted or placed in any location, and then you have some kind of uh, 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 essentially some kind of a cable to actually relay the reflected the image signal, and then onto a processing unit. In that processing unit, it generates the Raman shift, and then it can be sent to a AI platform, and then for the prediction of the AI. So this kind of, sorry, the prediction of the status of the conditions. So this is one example of the uh, application. The second application is the detection of the condition of the exosome. Exosome are some parts, some kind of internal materials secreted by, for instance, by stem cells. So those exosomes, they also have either they have medical purposes because the materials inside the exosome, they contain a lot of proteins, uh, important factors for the regulation of different systems like immune system, immunoregulatory factors, for example. So in this case, you can design some kind of array system. So on this plate, array template, you can actually essentially attach some kind of probe molecules. So those molecules can have the tendency to bind to a certain type of exosome. So you can see these are the exosome. Exosome, it has some kind of membrane with a simple inserted proteins. So if you have a certain molecules and a certain antibody, essentially it says here, essentially you use antibody as a attractor to attract exosome. Certain antibody only recognize certain protein on the certain exosome so that it can be specific to certain exosome. Once the exosome are attached on this array, and then you can actually shine the laser light, and then so for each array you get a Raman shift, Raman spectroscopy, and then and then you can use this Raman spectroscopy data to develop AI for the recognition of something, right? So, so it can be quite individualized. So each array can actually attract quite a number of exosomes. They are of different types. The types are dependent on upon what antibodies you use, right? So these kind of things. Okay. For this kind of instrument, equipment develop or apparatus development, not only you can develop the plate type of array, you could also develop this kind of three-dimensional nano structures that can actually hold the exosomes onto this kind of, there are some cubic type of unit that can actually attract the exosomes. So this way, the exosome is more because you have this specially designed unit, three-dimensional unit to hold the exosome. So thereby you can see, you can separate different exosomes more clearly to generate more clean or high quality, uh, more precision type of data, essentially. So the higher the quality of the data, the better the AI can be developed for these kind of purposes. Okay. So far we talked about the spectroscopic technology. You can see technology develop very fast. I didn't include some. I think there are some recent news reports saying that the Raman spectral meter device can be developed into a miniature type of things. At least it is of the size of just a hand phone, and then it can be even smaller. Just like you have this uh, laser pan, right? Laser pan can be it can emit laser beam with a small size as small as a pan, right? Just like that. Certainly, you can imagine a Raman spectrometer, which is also some laser emitted along with a cable. Uh, you can optical cable, and you can really develop a very very small device, wearable type of devices. With this kind of hardware development, certainly if we can combine the modern high-tech hardware with the new modern deep learning method, certainly it's better. So now we look at specifically how spectroscopy deep learning methods are developed. Okay. So this is an example. So we can easily view the spectroscopic pattern of these peaks and those kind of valleys, right? We can simply regard it as a one-dimensional data. So with the value of so this is a one, is just a vector. The value of each component of vector is simply the height of this wave pattern. By this way, you simply have this kind of one-dimensional vectors. We also know that spectroscopy data tends to have some local properties. Why it is? Remember that, for instance, we look at this Raman shift. So it goes from smaller shift to larger shift, right? So this kind of patterns. And certainly those that tends to have similar shifts, may have some kind of common features to make them either the uniformly smaller shifts or especially large shifts, right? For example, so the small shifts on this side, large shifts on the other side, right? So so this kind of local feature, perhaps it may be easily captured by a one-dimensional scene. What is a one-dimensional scene? Remember, when we talk about scene, we got used to a pixel, a picture, right? It is a matrix form. And then we have some smaller matrix acting as a filter to actually scan the whole image to grab the local feature. Now, because the things are become, they all of one dimension instead of two dimension, right? So certainly we can regard a one-dimensional vector as a shrink the form of a two-dimensional matrix, and likewise we can shrink the filter from two-dimensionality into one-dimensional vector. So this way we can use one-dimensional vector to scan the one-dimensional this kind of initial data. And then so this way you can actually produce a one-dimensional latent feature. So that's how a 1D scene works. So this is a full version of 1D scene. So the spectral signal, one-dimensional vector, and this is a one-dimensional filter. Depending on how many filter you use, you generate multiple feature maps. Remember that these feature maps, they are all one dimension, one dimensional. And then you can use max pooling. Remember that pooling help us to reduce the dimensionality. So you, for instance, you could pool four components into one. So if you do that, that means the size of the next layer of feature maps actually becomes just one-fourth of the original size. So you can see the size actually is reduced. So and then you can have next round of learning process until you think it's enough to capture to actually generate the good latent feature for you, and then you can apply, you can extend the latent features, and then you can apply either a shallow RNN, or you can use any machine learning method for whatever task you want. So this is an example. This is a general form of 1D scene model for any spectroscopy deep learning purposes. Okay. So here is actually the actual example of 1D scene published by some paper. So they actually use you can see that so if you consider input as one layer, they have 5 layers to actually learn the features of the spectroscopic data using this 1D filter scanning this process. So after 5 layers, you can see that they use the max pooling a few times to reduce the dimensionality. And then you can extend now you have 42 actually 42 of size 32 vectors and then if you extend that it becomes this long vector, and then it use 2 layers of the shallow neural networks to generate the outcome for you. So this is the one example of the application of using 1D scene for spectroscopy deep learning. So the next example is the use of UNet for the purpose. So you have this input, then you have this output, and then you want the output actually reproduce the input. So this is the feature learning process. Once it generates a latent feature, and then it goes back to the decoder. Remember that you know that this part is a decoder. The decoder part, in the end, it regenerates the image. That's the unit approach. And manifold methods have also been applied for spectroscopy deep learning. So here is the example, the Raman spectra of two samples. And then it does the data analysis of the signals, and then it uses the manifold approach for reordering of the data. It generates this kind of figure. Once so this figure actually corresponds to this, this kind of thing. And then you can input this figure, this image, instead of the original data. And then it actually goes through this scene architecture for the learning purposes. Okay. So the next we want to look at the specifically how spectroscopy deep learning is applied to biomedicine, to biomedicine field. So the practical application and reviewed by publications. So this one I have already shown you so now I give you a little bit more details. So this is for brain kind of brain neural network brain prediction. So essentially, so it is for deep learning the brain. So they didn't specify uh, which disease. So you can so it can be some disease. So I forgot which disease. So anyway, it can be some disease, either tumor or maybe other neural regenerative degenerative disorders, those kind of diseases. Okay. So in this case, they use the 1D UNet model to actually learn the spectra. The spectra are here, and then they try to see whether the AI can distinguish the people with the disease or the people without a disease. The people with this condition or the people with a different condition, something like that. Okay, so the next application is let's kind of see the title myself. Oh, not remove it. So so this is of course this so it says here so it is for the diagnosis of cancer. So it is essentially pancreatic type of cancer. So it is in pancreatic tissue. In this case, you have the contrasting Raman spectra of the tissue. Either it is a normal, so the yellow one is a normal, or it is a cancer tissue. So, so this is the comparison of the spectra. So we wanted the AI to learn the pattern to actually distinguish these two type of spectra. In the end, to give the right correct label. So for the first one, the label must be yellow. For this one, the label must be purple. So this is the case, and they utilized a 1D VGG. VGG is a very popular scene, but it is an earlier time scene because it is computationally not too efficient. But anyway, so it is a quite popular scene architecture. So we use this, actually the author actually used this VGG architecture for learning and prediction purposes. So this is the kind of thing. So once you have this kind of AI, so it has the application for the diagnosis purposes, and it could also have some applications in during the operation. You see the doctor here, a patient laying on this platform for the operational platform. Okay, so in this case, because Raman spectroscopic device can be made very small, and you can also use a optical essentially you can use this kind of optical cable for you can actually you can flexibly move the cable uh, and then to measure some parts during the operation. So it has this kind of operation. Of course, it can be utilized as in a laboratory setting for instance, for multi-omics analysis and for the visual observation of the. So that's the kind of application of a Raman. So Raman laser is actually not too strong in power, so it's not going to damage the tissue. So it's not damaging. It is also just it is also uh, non it can be non-invasive. Of course, if you want to do this brain tumor diagnosis or certain tissues, and then you may have to use a long some kind of needle type of device to get it. So that's only it is the best you can say it is some kind of minimally invasive. Certainly, it's not totally non-invasive, but for many applications, if the surface or near surface observation is enough, Raman spectroscopy technology can be used as some kind of non-invasive applications. Okay, so the next application is the uh, Raman combined with histopathology for cancer diagnosis. So essentially, this is the Raman imaging. So you have an image, and each pixel of the image corresponds to a particular position on the tissue. So that position can be shown by a laser beam, and then you can measure the Raman shift after of the due to the laser beam. So that means that you have an image of many pixels. Each pixel has a Raman spectrum like this one. So you can use this kind of data to develop the deep learning model. That's exactly what they do in these particular publications for distinguishing different. So of course, this is not really exact distinguishment of the spectral. Actually, comparisons are here. Comparisons are here. You can see uh, the difference where the difference actually lays. Okay. So this is the performance of the AI for cancer diagnosis and for other type of conditions, diagnosis of other type of conditions, including the bone dif, differentiations and so on and so forth. Uh, So now we look at the application of for cell phenotype identification. So we know that the cells, depending on what type of cells, it could be a tumor. And for tumor cells, not only they are all tumor, but also they may have different, uh, actually have some kind of a different potential outcomes. For instance, some tumors may be more prone to immunological treatment. Some may be resistant against the immunological treatment and so on and so forth. So this kind of condition of cells are called the cell phenotype, biological condition or biological state. So, so here, a manifold learning is here. Method is applied. So you have the original Raman imaging for different cells, right? And then you can analyze the pattern of the Raman spectra in the sample space. You generate this kind of distribution. In the end, you can apply manifold learning approach to reorder these peaks. So and finally, it generates this kind of 2D figure map, 2D figure for you of the application. Actually, there are two ways to generate the 2D figure. One is the one I just described. The other one is to look at the PCA of the samples. And then based on the PCA, you could also do this manifold re reordering thing to reorder the pixel into this one. And then you have two type of data. So you have the data of the normal processing to generate the latent feature. You could also have this uh, essentially you could have the just here the PCA generated the feature as the input, and then later on you can combine them together for deep learning purposes. So this is the performance improvement of manifold method, the orange one, and then the other method. You can see in many cases, the actually the orange one beats the purple one and then the blue one, right? For many cases. So this is an example of how a deep learning method, particularly manifold learning method, can actually improve the performance in terms of the phenotype of any cell. This is the examples. Okay, I guess now we completed the lecture part of today. So now it is the time for questions. Any questions?
Okay, thank you, Professor. Let me first ask question in a physical participants. Any question? Yes, ma'am, please come to the stage. Please first introduce yourself.
Hi, Professor. I'm
Hello.
Dr. Neha. Today I'm here again for you. I have two questions for you today.
Okay, yes.
Yeah. My first question, can we design a good and effective drug or ligand by knowing the structure of targeted proteins like TNF alpha, NF kappa B, or any other target proteins?
So there are AI methods for you to generate the molecules purely based on the essentially the structural features of the binding site. It could be possible. There are softwares, AI softwares to do that. Of course, people are concerned. Of course, you need a guide to guide the so it's easier to generate some kind of molecules to satisfy certain kind of essentially geometrical boundary conditions that can be relatively easily done. But you need to consider two other factors as a guide. One is that is the molecule designed synthesizable by humans or by any machines. So there's this kind of synthesizability issue. So there are something that can guide you to design the molecules. Another thing is that you want your molecule to be useful. Typically means the bioactivity. You want a bioactive molecule, so you have to have a activity guide. So essentially, you need either a deep learning model or any other model, even molecular docking, to help you to evaluate or to score the molecule design to see where, and then you use that score as a guide to select or to even to design subsequent molecules. Okay.
Okay, thank you for detailed answer. The next question, if I design any kind of drug or ligand targeting the structure, knowing the structure of the protein, whether it is authentic and effective for the rational use that we can synthesize in the lab further.
So normally, the normal practice that the initial round, I think just in one of my previous lectures, I mentioned a work published in Nature Biotechnology. So they have to manually evaluate the quality of the AI generated structure to select a subset of them for synthesis and for those kind of things. So in this case, I think in general, human intuition is still very very important based on the experience of established medicinal chemists, for example, and they can tell you whether the structure is really look like it is synthesizable, look like a bioactive molecule, those kind of things. And also there are certain substructures they deem to be less stable or even harmful. There are some toxic units, for example, and so some medicinal chemists they are very good. They are very experienced. They are very good at telling whether you can't have this subunit here because it is deemed toxic or it is unstable, easily leads to oxidation reaction or whatever those kind of processes. Okay.
Okay, thank you very much. So the last question. So today you have discussed about Raman spectroscopic deep learning. So is it applicable for the detection of proton NMR C13 and other spectroscopic analysis, which are normally we use in chemistry?
Of course, it can be utilized in any, as long as it's some kind of data. as long as the data of different samples, they have some difference, and in principle, AI should be able to detect that, to grab the differentiative feature to separate that. Okay.
Thank you very much. So this is the last one. Actually, right now we are writing a project regarding the synthesis of different heterocyclic compounds targeting different proteins. So with the help of AI, right now I'm learning from you the detailed lectures. So may I get your help? Me and my supervisor, we are writing the both. So may we take your help in designing the drugs targeting the different proteins, if you don't mind?
Yeah, we could. And so my suggestion is that so if you have the access of more than one AI, and also if you can employ non-AI method, try to combine them, combine the collective capability of those methods to help you to develop the kind of new structure you want. And another thing is that different AI, they obviously they are optimized. They were optimized under different circumstances, right? So if you for the particular target you have enough training data, for instance, enough known inhibitors, and you may try to use those inhibitors to further optimize the AI. Essentially, you actually slightly adjust the parameters to make it more suitable, more towards the task you really want, instead of a general one. So in other words, it means that if something is good for all purposes, it may not necessarily the best for a special case. So here you want a special case, and you can make some adjustment. For instance, in a special case, maybe they prefer to have this ion involving this ion interaction, then ionic interaction, then you need to have a more essentially more weight on the ionic interaction, for example. Okay.
Thank you very much. Thank you so much.
You're welcome.
Okay, thank you, Professor. Anybody else have a question? Okay. Hello, Dr.
Hello?
Chen.
Yes.
Okay. The next question is by Dr. Aisha, and she's asking, do we have any metal-sensitive sensors? In one study, you used AGNPs. We can use different metals to check toxicity.
We don't have that because we have not worked on those kind of things, so we don't know the specific one actually.
Okay. Okay, thank you, Professor. So, do anybody have any query to ask from our professor? Okay, thank you, Professor. I think it's all for today.
Okay.
We'll meet you soon. Tomorrow, inshallah. Okay. Bye-bye.
Okay, bye bye.